Okay, guys, we're going to go ahead and get started. Those of you in the Zoom and everybody here, thank you for coming. We're going to be going over the lecture material for exam 1. So it's covering chapters 1, 2, and 3. Not much to say. If you are on Zoom, we do have practice lecture exams and a practice lab exam available in Frio 111. So these are the chapters, like I said, to be covered. Chapter 1, kind of the basics, anatomy versus physiology, the differences of the two, body organization, homeostasis, directional terms, and the body cavities. Chapter 2 is going to be basic chemistry and biochemistry. And then chapter 3 is the cell and transport mechanisms. So exams should start for the remaining professors that haven't done their exams yet this week, tomorrow, which is Thursday the 13th. What was I going to say? Some of the classes didn't get to finish actually covering all of the material, so make sure you go back and read what you didn't finish in class. So besides like all of the directional terms or like the language of anatomy stuff in chapter 1, the big thing to know and to remember for the rest of the semester is homeostasis. So homeostasis is the process your body goes through to keep itself regulated. So the way that you can think about this is like always being like on, what do they call it, like a seesaw type thing. You always kind of want to be in that middle part because anything too low or too high can cause an imbalance in your body. Should have got the little thing. So this little image here, that may possibly come up on your exam. Just knowing like the imbalance, which is the stimulus, receptor, control center, effector, the response to change, and then that imbalance is corrected. I also like the image that's on the right-hand side with like the little wavelength thingies. And that's just how your body is staying healthy and staying in like a healthy state, not going too much far or below where it's supposed to be, because then that can lead to disease. And additionally, homeostasis does not involve keeping the condition static. So it's not trying to keep everything in like one line. It involves keeping conditions within tightly regulated physiological tolerance limits. So you don't want to keep your body like completely flat in a line. We do need those little ups and downs to keep our body within equilibrium. So variables are factors that can change within our body. So blood sugar, body temperature, blood volume, things like that. On your exam, I would maybe know the different factors that can change homeostasis or that can cause your body to fall out of homeostasis. And besides the temperature example, what is another example of your body regulating homeostasis? Anybody have an idea? What?
Positive feedback.
Positive feedback with blood sugar. Yeah, that's the big one, blood sugar. Keeping your body, again, in equilibrium. You don't want that too high or too low. I do know on your exam, there should be an essay question, and it will be, or there's going to be several. One of the options is going to be about homeostasis. So you may want to go into your exam review that's on Canvas. If you don't have that, please see me after. I can give that to you or email it to you. Go ahead and prepare that essay already, whichever one you do pick, but just so you can be prepared to already kind of have an idea of what you want to write on your exam. It does not have to be like a full-on essay, 3 to 4 paragraphs. You just want to be able to get your point across in, I believe it says something about homeostasis and then the difference between positive and negative feedback. Okay, and then looking a bit more at the receptor, control center, and effector. So the receptor is the sensor that responds to the stimuli. And if we go back, right, here's the receptor. It's kind of that first stimulus that changes. Does anybody know an example of a receptor? Something that kind of throws your body out of homeostasis. Maybe like a disease or an illness. Let's say you catch a cold, right? Your body then tries to fight it with the fever, and then eventually you come back down to homeostasis. So from the receptor, we move to the control center, which determines the body's appropriate response. So an example of a control center is your brain. The way this could come up on the exam is like describe or determine a structure in the body that acts as a control center, your brain. And then the effector is the response that either reduces or enhances the stimulus. So this is where we talk about positive and negative feedback. And I want to say what I'm about to ask usually comes up on the exam. So what is the most used feedback mechanism in the body? You got a 50% chance of getting it right. Positive? Anybody else? We'll say negative. So we have one positive.
Negative.
Negative.
It's negative
Mhm.
because when you're
Mhm.
contracts the muscles to bring heat to
this.
the
Mhm.
surface.
Yeah. So negative feedback is going to be the most commonly used in the body, and that's because it's kind of always going.
Wait, so a receptor is not exactly like something that receives something, it could be anything that throws anything off balance.
Yes, kind of. But usually it's like a switch. Mhm. So feedback loops, we have negative and positive. The most used feedback mechanism in the body are the negative feedback loops. And this is because it's kind of continuous. So it's always something going. It's a lot easier to bring something back down, whether to like keep it going with the positive. So remember, or is it? The effector is responsible for reducing or enhancing the stimulus. So in this example of the feedback loops, the negative feedback loop would be reducing the stimulus. So when something occurs in your body, whether that's, well, in the case of negative feedback loops, it's not always like super traumatic. The response your body gives off is to reduce or shut off that stimulus. So if you have, let's say, a blood clot, your body's response is to shut off that original stimulus, and it sends like different cells to that blood clot to kind of stop it. So the whole point of the negative feedback loop is to shut off the stimulus. So an example of this is regulation of body temperature and then the regulation of blood glucose. I want to say the essay question asks about an example for each of these two. So you may want to have an example ready to go.
Stated,
Yeah.
like for the positive, it was a tear in a vessel,
Mhm.
and then together
Yeah.
to repair.
I believe, yeah, you can use that one, and I believe that one's also in the book. So if you needed to go back and look at it, it's in the book.
Oh, so give me like a cut, like, like, let's say like, I don't know, like, they get a scratch
Mhm.
and there's like a response.
That would be the positive, right? For me, it's labor.
Yeah, the labor one is the like popular one with positive feedback loops. So the positive feedback loops, these are responses that enhance or exaggerate the original stimulus. These are usually infrequent events that do not require continuous adjustment. So the big thing to remember between the two is the positive feedback loops are more infrequent. And this is why, at least with Mr. Torres, he always talks about the going through labor example, because realistically, you're only giving birth at least once a year, every 9 months or so. So those are infrequent and don't always require continuous adjustment. And then, like I said, in the birth/contraction/labor example, the birth ends the stimulus and shuts off that positive feedback mechanism. So there you have one example for your essay question of positive, and then 2 examples for negative for your essay question. Okay, and really quickly, this specific image is more catered to lab, but they can ask you about anatomical directions in the lecture. So the way this can be asked is which directional plane cuts the body into left and right parts, the mid-sagittal plane. So as you're studying, kind of think about how this can be asked in both lecture and lab and kind of create your own questions. Or it may be in the, it may kind of be in like a matching question. So they'll give you the definitions and then you need to put the either median, frontal, or transverse, match that with the definitions. Every professor is different with their exams. So I can't say whether or not there will be pictures on your exam, but I would just be prepared.
Like it's like the little acronym, kind of just remember it. And I thought like, oh, like M.
Yeah, I do everything in acronyms because if I don't, my brain won't remember it. So when we get to like the fun stuff, which I say the fun stuff is bones, like all I have are acronyms and mnemonics, and you'll get annoyed with my mnemonics. Okay, no, why isn't this going?
So,
Oh, there we go. PMAT, yep. So the last thing I think I'm going to cover from chapter 1 are the body cavities. Again, this image is more geared towards lab, but you can get questions from the body cavities on your lecture exam. So they can ask you, you know, what structure is housed in the dorsal body cavity. So name one structure in that cavity. Let's see, we have our cranial cavity and our vertebral cavity. So if we just need to name one, brain. We need to name another, spinal cord, things like that. They may also ask what structure separates the thoracic cavity from the abdominal pelvic cavity, the diaphragm. So again, as you're studying, make sure you're kind of creating your own questions and figuring out how this can be asked in lecture and in lab. The other thing with the body cavities to make sure you go and take a look at again is the serosei. Identifying the serosei may come more in the lab, but you will want to know like the definition and the purpose of them.
So describing it,
Mhm.
would be that it envelops the
An organ.
organ.
Mhm.
So, because you have the visceral and the parietal. So the visceral will be inside.
Inside,
the
mhm.
parietal will be outside.
Outside. Mhm. Correct. And then, you know, the purpose definition, purpose, lubrication, right, for your
What
organs.
about quadrants? Quadrants.
On the lecture exam, it might ask you, so the question was if we would do quadrants and regions on the lecture exam, it may ask you like name an organ system in the right lumbar region or something like that. And that one would probably be like a multiple choice question. Okay, so now we are going to get into everyone's favorite part, chemistry. So I'm gonna kind of tell you like what you need to know for the exam. I know the book, the chemistry chapter itself is super long, and so are the notes. I think there's like 8 pages of chemistry. So I didn't put a whole lot of the extra information on the PowerPoint. I kind of just wanted you to know exactly what you needed to know. So the easy stuff, matter and energy, states of matter, that's what we are. We take up space, solid, liquids, and gas. Solids, these are constant, definite shape and volume, bones and teeth in the body. That's how that relates to anatomy. Liquid, a definite volume, blood plasma in our body. And then gas is neither a definite shape or volume, and that's oxygen. On the exam, this could possibly be like, which of these is a solid found within the human body? Bones. And then forms of energy are also important to know. So I've been telling students the book has everything you need to know. These sets of notes that some of the professors have on Canvas kind of break down, they break it down into simpler terms. Not everybody has access to these, but I can send them to whoever needs them. Just make sure you see me after. So these are our different forms of energy. I'm going to go through them one by one. Kinetic energy is energy in action. So when we're moving, riding a bike. Potential energy is stored or inactive energy that has the potential to do work. So it's in the name, it has the potential to be productive, basically. So in our body, does anybody know what what is a potential source of energy?
Fat, right?
Fat. One of the big examples is ATP in our body, right? So that's a potential source of energy. Chemical energy stored in the bonds of chemical substances. So, right, energy we get from eating foods, and then we turn that into kinetic energy when we take a walk, when we go for a run, when we ride our bike.
Wait, how many tests I got electrical and radiant? They're
Mm,
kind of radiant waves.
yeah, radiant
I think it
waves.
was starting off with the questions,
Mhm,
the examples he gave, and I got both of them.
yeah. If you may want to put a star, and I can look at it after,
Okay.
because I haven't seen the questions.
Okay.
Electrical energy, the movement of charged particles. So in our body, our nervous system uses electrical currents to transmit messages, right? So our brain telling us a part of our leg hurts, our heart using electrical energy to contract. Mechanical energy is energy directly involved in moving matter. So lifting weights. And then lastly, we have radiant / electromagnetic energy. This is energy that travels in rays. So visible light, infrared, radio waves, and UV waves. For your exam, I can see this being asked, like riding a bike is an example of what type of energy. Stored ATP is an example or is a potential source of what type of energy, potential energy, different questions like that. So I would know the definition for each of these. Like I said, the book goes into like super niche detail of all of them. This breaks it down into what exactly you need to know. And if you don't have it, see me. Okay, the next thing that's big to know is 65% of our human body is made up of one singular element. Does anybody remember which element that is? Which element makes up 65% of the human body?
Oxygen.
Oxygen, yeah. So oxygen makes up 65%. The other, 31% of that is carbon, hydrogen, and nitrogen. So like I said, I have to break everything down into like little mnemonics or little acronyms. So that's just how I remember the 4 elements that make up majority of our body. The way this can be asked is, which element is most abundant in the human body? Oxygen.
Oh, okay.
I don't know if they'll ask, they usually don't ask for like specific numbers, but a number that can come up is like 96% and then probably 65, just because 65% of us is oxygen. Kind of in this same realm, now we get into like isotopes and stuff. So I would know the difference for each of these. Again, I haven't seen the quiz questions, but I feel like someone brought something similar up to me. So isotopes have the same number of protons and electrons, but vary in the number of neutrons. Radioisotopes are still isotopes, but they're unstable and they spontaneously decompose into more stable forms. So this radioisotopes are used in like the biological research and medicine, things like that. A molecule is a combination of two or more atoms held together by covalent bonds. And then a compound is when two or more different kinds of atoms bind together. So the way these can show up on the exam is, again, they'll give you all of the definitions in blue in your multiple choice, like those are the answer choices. And then the question will be, identify the definition that's being talked about or something like that. And they'll give you, have the same number of protons and electrons, but vary in the number of neutrons. You would need to select isotopes, radioisotopes, molecules, or compounds. There we go. Okay, so mixtures, there's a really good video on, I put Blackboard, I meant Canvas. The video on Canvas that is provided to you. If you can't find those, please see me afterwards because those are very helpful for not only the lecture, but also the lab. It breaks down what mixtures are and what exactly you need to know on the exam. I will say for the lab, sometimes they pull the lab questions straight from those videos. So you are going to want to make sure you're looking at those videos. But mixtures, right? We have three different types of mixtures. Solutions, these are homogeneous mixtures, and these may be solids, liquids, or gas. So with solutions, we have solvents and solutes. Solvents are usually the substance in the greatest amount. So what is like what subject there's more of or what there is more of. And there's usually something that's always a solvent. Does anybody remember what that is? Water. Yeah. Water is usually always the solvent. And then solute is the substance in smaller amounts. And then we have colloids. These are also called emulsions. So if you haven't seen that in the book, it may come up as emulsions in the lab. I'm sorry, not the lab, the lecture exam. So just know that that's the same thing. These are heterogeneous mixtures that often appear translucent or milky. So a real life example is Jello. In your human body, it is the cytosol inside of the cell. The way this can be asked is cytosol in a cell is an example of what type of mixture? A colloid. And lastly, we have suspensions. These are heterogeneous mixtures with large solutes that settle out. So an example is blood in the body or sand and water. And real quick, yeah. So we have the example of a heterogeneous mixture, sand and water. The sand settles at the bottom, whereas with the homogeneous, we have sugar and water, and it turns into sugar water.
Oh, so yeah, okay, so one can be taken apart, the other one really can't.
Right, like you wouldn't be able to go in the sugar water and pick out the sugar, but with the sand, you could probably grab a little, thumbful and have that. Go back. So again, with mixtures, make sure you go and watch that video. That would be really helpful. Next, we're going to talk about chemical bonds. So the first one is ionic bonds. These are electrons that can be transferred from one atom to another. And then after this, ions result when atoms have lost or gained electrons. And so the atom that loses that electron acquires a positive charge and is a cation. Yeah. Again, you would probably want to know like when an electron is transferred to or from one atom to another. That's usually an ionic bond. And then what results after the electron is lost and put into another one, and then acquires that positive charge, that's the cation. And then we have covalent bonds, which are formed when atoms share electrons to fill those outer shells. And this satisfies the stability needs of each atom. Another big thing to know with the covalent bonds is they may be either polar or nonpolar. So that could be like a true or false question. So it could be like true or false, covalent bonds are only nonpolar. False, because they can be polar or nonpolar. And then we have hydrogen bonds. These are formed when a hydrogen atom is linked to another atom. Usually these are nitrogen or oxygen. And so when the other atom is attracted by another electron-hungry atom and forms a bridge between them. So the big thing about the hydrogen bonds is they are weak and easily broken. So on your exam, what type of bonds are easily broken? Hydrogen bonds. Another big thing to know with the hydrogen bonds is DNA and proteins rely on these types of bonds to take on their 3D shape. Okay, and then there's that mixture picture again. There's also like images on Google that kind of break down mixtures as well. For some reason, these didn't make sense to me at first, but they make sense. Okay, now we're going to get into cells. Real quick though, back on chemistry, you're gonna wanna make sure you go through the pH scale. So true or false, the pH scale is measured from 1 to 14.
False.
False.
False, it's 0 to 14. Yeah. So on this, yeah, remember on your exam, 0 to 14 is the pH scale. That will almost pretty 99.5% sure that's going to be on the exam.
They're kind of tricky.
Mm,
The synthesis and decomposition.
yeah, probably. I didn't have enough time to put it in here, but I would say it'd be more like these right here. But we can go over those as well. Let me see anything else from You're also going to want to know the monomers for some of the compounds. Those should be in your textbook. Again, I don't have all the time to go over everything, but if you do want to go over something specific, see me.
Like, nucleotides.
Yeah.
Nucleotide, amino acid, protein amino acids.
Exactly. No. Okay, so now we're going to get into chapter 3. I know some classes didn't get all the way done with chapter 3, so just bear with me, try to get through it. So cells, right, they're the structural units of all living things. Cells only arise from other cells. This is where we get mitosis, which we'll talk about in a little bit, but that's kind of one of the bigger pictures of this chapter. And then mitosis, right, like a simple definition, division of the nucleus. And then another big thing to talk about is the plasma membrane that surrounds the cell. So the membrane is selectively permeable. What do we think that means in our own simple words? Selectively, right? So it lets things in and out selectively. It wants certain things in, it wants certain things out, it doesn't want certain things in. So it selects and restricts the passage of substances into the cell. The transport of substances, so whether that comes in or out, depends on various factors. Lipid solubility, size of the particles, electrical charge, and the presence of channels and or transporters. I can see this being like a select all that apply question, or maybe finding the one that doesn't belong in the question. So, select or which of these is not a factor for transport of substances? We can say the color or something like that, right? Something silly that wouldn't make sense. But definitely know these 4, lipid solubility, size of particles, electrical charge, and the presence of channels are factors that are dependable factors for transport of substances. Okay, and then membrane transport processes. So we have the passive transport process. Does this require energy or no energy? No energy, right. So passive, think of it like you're doing something in passing. It's something really easy. You don't really have to do a lot of work. No cellular energy required. This is the net movement from an area of higher concentration to an area of lower concentration. So higher to lower for the passive transport process. Next is diffusion, and I don't have it on here, so I'm just going to kind of read the definition. The net movement of substances down the concentration gradient. So with diffusion, the membrane must be permeable to the substance, therefore it's nonpolar. So again, diffusion is still a passive transport process, and this is the net movement of substances down the concentration gradient. And then we have facilitated diffusion / carrier mediated. I want to say you could call this either or on the exam, or you can see it written either or. Facilitated diffusion is when certain molecules, such as glucose, move across the membrane with the help of a carrier protein or transporter. And these carriers transport specific substances.
You said diffusion was passive transport, or it was?
It's a passive, yeah. So again, the facilitated diffusion, it's called facilitated because they have the help of a carrier protein. So again, what type of transport process has the help of a carrier? Facilitated diffusion. And then osmosis, does anybody know what like the big difference between osmosis and diffusion are? The water, yeah. So osmosis is the movement of water through a selectively permeable membrane. So again, very selective with what they let in, but in this case, we're talking about water. And then with osmosis, it's from an area of higher water concentration to an area of lower water concentration. Correct. And then I know my class was barely getting here today, but this is an image in your book, and I want to say they use blood as an example. But this is one I just found on the internet. These are the types of solutions. So we have iso, hyper, and hypo. So those are like the three prefixes you're going to want to remember. And then tonic, those all just mean strength.
Sucks out the water
Mhm.
and then it shrinks.
Yeah, like it creates it. Mhm.
I know those terms. that we need.
I don't think you'll have to know them. You should just like be familiar with them because they may come up like in the question. So let's talk about an isotonic solution. It contains equal amounts of solutes as those found in the cell. And with isotonic, there is no net movement of water. So, or break that down even more, isotonic, there's the same concentration of solutes and solvent. So we can see it here in this picture. The solvents are these little blue dots and the solute are the yellow. There's the same amount of them. Again, so iso, same. Hypertonic, more. So with hypertonic, there's going to be more solutes and less solvent. So with hypertonic, this is when water leaves the cell, causing it to shrink or crenate. This was if you are in Mr. Torres' Wednesday class, when he started talking about the grape, you take all the water out, turns into a raisin, crenates or shrinks. And then hypotonic, so hypo, less. There are fewer solutes and more solvents. So in hypotonic solutions, they contain less amount of solutes as those found in the cell. Water moves into the cell, causing them to swell or burst. Another word that they use is lyse, L-Y-S-E, burst. So just in case you see that on the exam, lyse means burst, and then crenate means to shrink. Again, there is a specific example in your textbook, should be in chapter 3, but it does also break it down even more. But make sure you know these 3 types of solutions. Again, you really only need to remember the prefix, so iso, hyper, hypo, and then tonic for all 3 of them. Okay, and then we move into active transport processes. So we just talked about passive. Now we're going to talk about active. So active transport processes are dependent on ATP or adenosine triphosphate use by the cell, since substances are transported against the concentration gradient. And then, so again, these are all active transport processes. we start with the primary active process. So these are also called primary active / transport sodium / potassium pump. That's a huge, long name for something if you want to break it down even more. So this is, again, an ATP-driven pump that requires, obviously, sodium, potassium, and then ATPase, which is an enzyme. When this primary active process is occurring, it pumps 2 potassium ions in and 3 sodium out. So I want to say I've seen of exam questions like asking about the specific numbers. So how many potassium ions are coming in, how many sodium ions are coming out? Mhm. So these ions are coming out to ensure that the diffusion of these ions will never reach equilibrium. So they will never be relaxed a day in their life. Okay, and then we also, or next we have secondary active transport, or also called a co-transporter. This is the transportation of other substances dragged either in the same direction or the opposite direction as a result action of the sodium-potassium pump. So again, a way this can be asked, secondary active transport is an example of what type of a transport process? Active instead of passive. Okay, and then we have vesicular transport. And then the two parts of this are exocytosis and endocytosis. So anything we do in anatomy in this class, exo or X will always be leaving, exiting. Endo will always usually be coming in. So exocytosis is when substances are removed from the cell. Example is secretion of antibodies by plasma cells or the release of neurotransmitters from neurons. Endocytosis is substances brought into the cell. So again, we have that plasma membrane. Again, it can either be selectively permeable, it can just let things in, but this is where that endocytosis comes in. And then with endocytosis, we have phagocytosis, which is cell eating, and then pinocytosis, which is cell drinking. The cell will take in small droplets of intestinal or interstitial fluid containing dissolved solutes. So for this, the way this can be asked, phagocytosis is an example of what type of vesicular transport? Endocytosis. It's a lot of cytoses. Like the definition or the question? I forgot what I said. I think I said is a type of
Phagocytosis was an example of endocytosis, right?
yes. Or what did I say? I already forgot the question. I think I said phagocytosis is what type of vesicular transport, something like that. And that would be endocytosis. Or is a part of what type of vesicular transport? Okay, and then the last one we're going to cover is the resting membrane potential. In the resting state, all body cells exhibit a membrane potential of -5 to 100 millivolts, and as a result, cells are polarized. The big thing to remember here is -5 to 100. This is another one of the numbers that you need to memorize. I think on the exam, it'll ask you like what the membrane potential is, and then it'll give you like different numbers. So -5 to 100 millivolts. Okay, and lastly, yeah, we're gonna go over like parts of the cell really quickly because there's just specific things that you need to know. I thought I had my pointer, but I don't, so we're just gonna go through it. First, the cytoplasm. So the cytoplasm is the gel inside. We call all of these little structures cytoplasmic organelles, which mean like little organs. These are all specialized cellular compartments with specific functions to conduct everyday life, different things. Oh, thank you. Perfect. Some of these are non-membranous, so they have no membrane, but most have membranes. There it is. So the first thing you learn in like third grade science, what is the powerhouse of the cell? Nothing exists, mitochondria, yeah. So the mitochondria are the powerhouse of the cell. These are the sites of cellular energy production, right? So mitochondria, energy production. Then we have our ribosomes. Sorry, on someone's head. These are the sites of protein synthesis. And then we have two types of endoplasmic reticulum. A lot of the time you'll just see it as ER. That's what it's referring to. So we have rough up here and then smooth. Rough and smooth. So rough ER, those have ribosomes that synthesize the protein secreted from the cell. So again, rough ER, those are rough because those have ribosomes. You can't really see it on this one, but if you go back there and look at our cell model, you can easily distinguish which are rough versus smooth. And then smooth ER, these are responsible for lipid metabolism, cholesterol, hormone synthesis, drug detox, and the breakdown of stored glycogen. We have the Golgi apparatus right here, also called Golgi bodies. Kind of a weird image of it. Again, it's a lot better back here on our cell. It's this pink guy right here. The Golgi is responsible for packaging, modifying, and secreting proteins and lipids. It's kind of like the mail carrier.
One more time.
Yes, the Golgi apparatus or Golgi bodies are responsible for packaging, modifying, and secreting proteins and lipids. Lysosomes, right here, right here. These contain enzymes that can destroy foreign particles. Okay, and then we also have cytoskeleton filaments. Those are not on our image here. I want to say you can see them back here. If not, I know the image in your worksheet definitely has them. These are series of rods which act, like as they say, as the cell skeleton. So it works as like skeletons, muscles, and ligaments. No, the cytoskeleton filaments. And then the three structures are the microtubules, the microfilaments, and then there's also intermediate filaments. So the microtubules determine the overall shape of the cell. Microfilaments are involved in changes of the cell shape. And then the intermediate filaments act to resist pulling forces on the cell.
That are involved in changes.
Microfilaments. And then the last one is centrosome and centrioles. I don't believe they're on here. They are back here. That's this little black circle right here, the centrosome. The centrioles are the yellow, they kind of look like, honestly, little pasta noodles that are coming off of there. These are very important during mitosis. So during mitosis, the centrioles work in separating the cellular contents, which then produce, right, the two nuclei, which then turn into the two daughter cells. I know we didn't cover, I don't think, any mitosis with the cells. Just going over, right, the different stages and then what's occurring at each stage will be sufficient for the exam. Knowing the definition of mitosis, and then knowing the, like, what's occurring during, let's say, prophase versus what's occurring during, like, cytokinesis. And then understanding what a daughter cell is. What is the purpose of mitosis? Anybody have like a short answer? Reproduction, multiply. Yeah, cell growth and reproduction. So if they ask you, what is the purpose of mitosis? 4 words: cell growth and reproduction. Right, so when you're answering questions on the exam, if it's short answer, that's what they're looking for, is a short answer, not a whole sentence, right? Because most of the professors have like 100 or so exams to grade. So make sure you get your point across, but don't write a novel. Okay, and that was it for the review. Those that are in Mr. Torres's sections, don't forget that you'll be taking your exam on your laptop. So make sure you go to class with it fully charged and ready to go, connected to the Wi-Fi. Make sure you're on time because you only have the, what's it, hour and 15 or 75 minutes to take the exam. And then for the next week or so, or maybe it's even tomorrow for some of you, focus on the lecture portion of class, and then immediately following your exam, go into your lab portion stuff. Like I said, depending on the professor, there's not usually a lot of images on the lecture exam, but they can still come up. So if you have any questions, feel free to stay behind. Those of you in the Zoom, we do have, like I said, the practice lecture and lab exams available in the lab. Come see me if you need any help, or we also have Julian, Joey for anatomy.